First off, let me introduce myself. My name is Claudia van Wyk. I'm the project coordinator today, and I'll be guiding you through the introductory slides. Following that, we will have presentations from our esteemed presenters, who I will provide more background on in a moment. We will be hearing their insights over the next 25 minutes, and I really hope you are as excited as me to hear and learn from them today. After the presentations, Bethany and Cheryl will facilitate a dynamic discussion, and this will be an excellent opportunity for everyone to engage in meaningful dialogue. Finally, we'll open the floor for a Q&A session, so please keep your questions ready. We are looking forward to a productive and engaging session with you all. So the Initiative for MPTs, or IMPT, has been a cornerstone in the global effort to advance multipurpose prevention technologies since its launch in 2009. Since launching, the IMPT consists of 2,000 stakeholders from nearly 24 countries. This unique initiative acts as a neutral platform, bringing together stakeholders from various backgrounds to offer essential technical guidance and strategic direction. By uniting diverse voices, the IMPT works tirelessly to push forward the development of MPTs, which are crucial for HIV prevention and sexual reproductive health. In 2020, recognizing a need for ongoing dialogue, the IMPT introduced the Let's Talk MPTs webinar series. This series has successfully gathered global experts to discuss research and development priorities, and also fostered collaboration and networking among leading figures in the field. So as we dig deeper into today's session, remember that these efforts are paving the way for innovative solutions in sexual health and prevention. It's my honor and privilege to introduce today's presenters. We have Dr. Lisa Rowan. She is a professor in the Department of Pharmaceutical Sciences in the School of Pharmacy. She also holds appointments in the Department of Obstetrics, Gynecology, and Reproductive Sciences in the School of Medicine and Clinical Translational Science Institute at the University of Pittsburgh. Additionally, she is an investigator at the McGee Women's Research Institute. She obtained a Bachelor of Science in Chemical Engineering, a PhD in Pharmaceutical Sciences, and completed a postdoctoral fellowship in mucosal immunology. With over 35 years of experience in pharmaceutical product development, including several years in the pharmaceutical industry, her current research focuses on the areas of women's health, infectious diseases, and global health needs. Her laboratory is dedicated to transforming global and women's health through pharmaceutical innovation. Her team has successfully developed over 50 pharmaceutical products, with 13 of them translated from the bench to the clinic. We also have Dr. Shravan Patel. He is an associate professor at the University of Pittsburgh and an investigator at McGee Women's Research Institute, with over a decade of distinguished experience in pharmaceutical product development and translational research. He earned his bachelor's in pharmacy from Birla Institute of Technology and Science in India, his master's in pharmacy from the University of Georgia, and his PhD in pharmaceutics from Duquesne University. As a technical lead and investigator of multiple product development projects, Dr. Patel brings exceptional expertise in formulation development across diverse dosage forms, including nanosystems, thin films, microparticles, and lipid-based systems. His work spans areas such as HIV prevention, contraception, and inflammatory diseases. His comprehensive experience spans preclinical pharmacokinetic studies, GLP toxicology studies, GMP scale-up activities, and regulatory submissions for IND applications, making him a recognized leader in translating innovative therapeutic products from laboratory concept through to clinical development. Thank you so much for being here. I will hand over to Dr. Lisa. Thank you.
Thank you very much. And thanks so much to Bethany, Claudia, and Cheryl for inviting us, and an IMPT for inviting Siobhan and I to present this webinar to you. Our group is involved in developing multi-purpose technology products, and so we're very vested into this effort, and really see a potential for these products to have a significant impact on women in the future. We are hoping to discuss with you some of the concepts around evaluating MPT product performance using experimental and computational modeling approaches. Next slide, please. So I'm sure many of you have seen this slide available from IMPT, but what's important to recognize is the development of multi-purpose technology products actually acknowledge this connection between women's sexual and reproductive health needs. And these are interlinked, as we know, and developing products that can target multiple indications actually are beneficial to women from an acceptability and a usability standpoint. Next slide, please. So just in reviewing the MPT landscape, we're seeing a number of MPT products being developed, and those products can take different shapes or forms. So we've seen vaginal rings, implants, injectables, oral products, IUDs, films, inserts, and gels for vaginally applied products. And also the indications range from HIV to protection against unintended pregnancy and other sexually transmitted infections, including gonorrhea, chlamydia, BV, and HSV. Next slide, please. So from the IMPT website, what we see is there are a number of actives ingredients that are being developed, and this is a list that's available on that website. And what you can see from the IMPT website active ingredient list is that you see that we have a very diverse number of compounds that are available. These compounds range from small molecules to macromolecules, including complex molecules like antibodies, polymers, dendrimers. And also they cover a wide range of therapeutic indices: gonorrhea, herpes, chlamydia, pregnancy, HIV. Additionally, the vaginal intended dosage forms are including multiple types of dosage forms: rings, gels, inserts, and films. So this really leads to a complex type of dosage form and complex environment that includes not only multiple actives, but also drug device combinations in some cases. So ultimately, those actives can also lead to different targets for action. So when we're trying to talk about drug targeting, especially in compounds that have multiple actives, we may have multiple targets as well. So this sort of illustrates the complexity of the MPT product itself. Next slide. So this complexity actually leads to some challenges across the drug development process. So these challenges include challenges at the preclinical, clinical, and the regulatory levels. In the preclinical state, as I said, these are complex products that are being designed. You have, in some cases, multiple actives, so we have to think about compatibility in those complex dosage forms. And there is a lack of predictive models that are available to facilitate that preclinical work that's needed. From the clinical aspect, especially those MPT products that are incorporating multiple actives, when we increase the number of actives, we don't just double the amount of clinical burden, we actually exponentially increase that. So with multiple actives, we have much greater cost, much longer timelines, and much more complex clinical trials that are required to be completed. And if we think about the regulatory aspect, now we have these two or more compounds, and those may be a biologic and a small molecule. You may have a device, so that's going to span different regulatory bodies that may have different regulatory bases for review of documents. Additionally, there's a lack of guidance and guidelines that are generally available in this area, which also serves to provide a challenge in advancement of such products. Siobhan, I'm going to hand it. And so in order to address those challenges, we are using experimental and computational models. And so I'll hand it over to Siobhan to discuss some of those.
Thanks, Lisa. Again, good morning, good afternoon, good evening. I echo Lisa's words there. MPT has been an excellent resource for all of us who's working in MPT, and thank you very much for your consistent and relentless advocacy for MPTs. We share that. So going back to the presentation, the predictive models can be divided into experimental and computational. And we have been working in this space, developing models on the in vitro, ex vivo side, in vivo, as well as in silico. For example, on the in vitro side, we have developed in vitro release methods that can that are biorelevant and can predict the differences between a very quick releasing product versus a slow releasing product. We have also developed mucoadhesion methods using cervico-vaginal tissues from women, as well as synthetic tissues, again, discriminating a product that is more mucoadhesive versus less, because that influences performance in vivo in the vagina. We also developed cell and tissue models to investigate toxicity, efficacy, such as anti-HIV activity, metabolism, uptake, and as I mentioned, formulation performance. So a lot of this data goes into IND applications. In the in vivo space, we have utilized, there are macaque models, rabbit models, and sheep models that are investigated for safety, efficacy, pharmacokinetics, and retention. And this data then feeds into in silico approaches, such as the non-compartmental analysis, which is a very traditional way of looking at pharmacokinetics data, to more advanced physiologically based pharmacokinetic modeling to predict pharmacokinetics and also scale animal data to human exposure, as well as predict the population-based variabilities. So we're going to talk a little bit, I will talk a little bit about the macaque model and how we utilized it in our film development. And Dr. Rohan will talk about the PBPK approach. So the female pigtail macaque model, also called Macaca nemestrina, has been widely employed in infectious disease research, especially in microbicide work, evaluating pharmacokinetics, safety, and pharmacodynamics. This model is, although smaller in size, it's very similar to humans in terms of anatomy, very similar vaginal epithelium, very less keratinized compared to other animal models, similarities in microbiota and also immunological profile. Although the pH is slightly basic, this particular aspect is very hard to recapitulate in animal models. So we have developed a large number of studies and methods using this model. And here a shout out to Dr. Dorothy Patton's lab from University of Washington, who worked with us in developing some of these methods, and also employed them in our IND applications for drug products. For example, the extended safety and pharmacokinetics up to 60 days was evaluated using this model. We have also developed coital studies where monkeys are trained to have sex on demand, so we can evaluate the impact of sex on drug distribution. Also developed imaging-based methods such as the MRI to understand dosage form distribution in the vaginal tract, colposcopy to visualize the presence of the product and mucosal safety. And here is an image showing the cervical surface in a macaque, very clean, no evidence of inflammation. At 3 hours after the product is inserted, you can see the presence of the product. And then over time, as the product is disintegrated and dissolved, you can see a less amount of that. And finally, going back to the baseline on day 20 without any evidence of film present or toxicity. So this model has been very critical in our evaluations and design of the film product. Also developed procedures for collecting tissue biopsies and other types of samples and evaluating safety parameters such as microflora, impact on microflora, cytokines, and immune cells. This model has in fact been, as I mentioned, critical in design and evaluation of a large number of vaginal films in our group. ranging from the quick dissolve dipivirine and tenofovir films to more recent combination products or MPT products. So I'm going to talk a little bit about sort of like an example of how we utilize the pharmacokinetic information from this model to inform and optimize our film, MPT film. So first, let's look at what a film is and how it works. It's a pharmaceutical product category. Film is a very thin and flexible continuous matrix or a sheet that carries the drugs either in a dissolved format or dispersed. It measures roughly 5 centimeter by 5 centimeter, and it's very thin, around 50 to 200 microns. And also it weighs very low, less than a gram, typically half a gram. So this is important because it has very minimal effect on the innate factors in the vagina because of this low weight. The films have been investigated and are also present in the market for oral use, such as the breath mint strips you might be aware of, as well as pain, orally disintegrating films for pain. We are employing vaginal films, films for vaginal application because of the listed advantages. Film dosage form being very small as well as thin can be a discrete dosage form. It's women controlled in the sense it can be self-inserted without the need for an applicator. It's also scalable, low cost, biodegradable, so it doesn't really produce waste apart from the wrapper that it is packaged in. And it can also lead to complete drug release. So these are some of the differentiating factors compared to other dosage forms that makes film an advantageous dosage form. There are not many products on the market. You might be aware of the vaginal contraceptive film containing N9, but the drug-loaded films for vaginal use are not marketed, at least in this country. So our enthusiasm for films development for MPTs is also shared by stakeholders, stakeholders in South Africa, Zimbabwe, and Kenya. When we approached them, they showed really high desire for combining contraception and HIV prevention onto this platform. And you can see an overwhelming number of stakeholders actually supported the MPT development and mentioned that it is a priority for the field. So this data was collected in one of our earlier studies under Matrix program. Similarly, in a different study, when women were asked to pick 4 MPT products, these were hypothetical concept profiles, you can see films did very well with respect to the preference. And obviously, women prefer different products. That again talks about the choice that we need to have. So this is in support of our using our film dosage form for MPT development. So with that, we have 3 products, MPT films in development in our group. The first one is dipivirine and levonorgestrel, previously supported by USAID under Matrix program. The monoclonal antibody combination film supported by NICHD, and EFDA, also called islatravir and levonorgestrel combination film under Latch program supported by NIID. I'm going to talk a little bit more about the EFDA and LNG film. So EFDA LNG MPT film was being designed as a 1-month product to release both the drugs islatravir and levonorgestrel. Islatravir is a non-nucleoside reverse transcriptase translocation inhibitor, highly potent, and it has really long half-life, which is very much suitable for long-acting products. And levonorgestrel, as you all probably are aware, is a widely used progestin contraceptive in several marketed products. So it was obvious for us to combine these two into an MPT film. However, there were challenges. Levonorgestrel is hydrophobic and EFDA is hydrophilic. So achieving that sustained drug release for both the drug molecules from the film is challenging. So we overcame this using innovative design strategy here as tri-layer film, where the outer layers act as a rate controlling membrane for the drug molecules, especially EFDA. And you can see a cross section of a tri-layer film shown here in the SEM image, scanning electron microscopy image. So in developing and optimizing this product, we really tapped into the pigtail macaque model. And here is a typical study designed for an animal study. Female pigtail macaques will be inserted with a film on day 0, and they are monitored for a period of 42 days. On day 30 or 31, if there are any remnants of the film, we remove them, and then we track the pharmacokinetics of the drugs after the film is removed also. We collected several components, blood components, plasma, PBMCs, dried blood spots, vaginal fluids, and also colposcopy to monitor the presence of the film, as well as to monitor safety. So these were the analytes that were measured: EFDA in swabs and plasma, LNG in swabs and plasma, and then triphosphate levels in PBMCs and DPS. So EFDA triphosphate is the active metabolite that is converted intracellularly from EFDA. So I'm going to show the plasma data because that is what was used to optimize the formulation, inform formulation development. So the pharmacokinetic data collected, or the drug data from plasma, was analyzed using a method called non-compartmental analysis. It's a pharmacokinetic analysis method. It generates very important PK parameters, and this is an empirical model, does not make any assumptions. It's all equations based. It generates Cmax, AUC, T-half for plasma. If you look at this graph, it's a typical plasma drug concentration versus time graph. As time progresses in animals, there is an increase in absorption of the drug into the plasma, and you hit a Cmax, that is the highest concentration in the plasma. And after that, and the corresponding time point is called Tmax, and after that you see a drop in the drug amount in plasma because of elimination. And we can fit rate equations such as first order kinetics to obtain T-half, which is the half-life of the drug in plasma. So these are all important parameters because if we want to achieve sustained release, then we need to have the Cmax pushed out towards the right, as well as increase the elimination half-life. So looking at this graph and understanding PK parameters become important in optimizing the formulation. So this is how we iteratively developed and optimized the product. Initial formulation contained low dose of EFDA and LNG in a tri-layer format. And we investigated that in the macaque model. You can see some of the PK parameters. I'm not going to overwhelm you with numbers, so let me just quickly show some important ones. You can see Cmax here for LNG is 0.21 nanograms per ml, and we were targeting about 0.35 nanograms per ml. So very clearly, even at the highest concentrations, we did not achieve the target for LNG, and similarly for EFDA. So we had to go back and redesign the formulation. We modified it, we changed the layer design, and also increased the contraceptive dose to increase the exposure. When we conducted the animal study, we can clearly see an increase in Cmax and an increase in exposure, overall exposure for levonorgestrel as well as for EFDA. However, this still did not meet the target for 30 days. So finally, we went back again and redesigned the formulation, this time increasing both the drugs. And also we realized that we need to increase the T-half, extend the T-half further in order to achieve that 1 month release. And that's the lead formulation. You can again clearly see an increase in Cmax, but also an increase in exposure for both the drugs with a simultaneous increase in T-half. So that means we are extending the elimination half-life, thereby achieving the targeted drug concentrations for a period of 1 month. And here are the PK curves for levonorgestrel and EFDA. You can clearly see drug levels are present above these target lines for a period of at least 30 days. So this is a very simplistic modeling approach of non-compartmental analysis using the pigtail model, but it has been instrumental in optimizing our products. However, there are gaps. There are gaps in traditional PK modeling. Non-compartmental analysis does not provide any information on distribution kinetics, and the prediction capabilities are limited. As I mentioned, we looked at the drug in plasma, but not all drugs are absorbed into the systemic circulation, such as proteins. And some of them are not correlated with the tissue or vaginal fluid levels, which could be our ultimate compartment for pharmacodynamics. So how do we predict drug exposure in vaginal fluids and tissues? Additionally, the cost and complexity of running animal studies, although they are required, we need them to understand PK, but the cost and complexity does make it challenging. And also there is no consideration for population level variability when you're looking at these empirical models, because we are not really accommodating any anatomy and physiology into these models. So the solution for this, to address this problem, is through physiological modeling. And I'll quickly introduce this. Physiologically based pharmacokinetic modeling, also called PBPK modeling. It's a mathematical approach, a framework to describe and predict the ADME properties, which is absorption, distribution, metabolism, and elimination of a drug. Unlike the empirical model that I talked about, a PBPK model takes into account different organ systems, their own properties such as blood flow, tissue permeabilities, and then combines that with drug-related properties and develops a mechanistic model using ordinary differential equations. This is grounded in biology because it takes into account the anatomy and physiology. So what kind of data goes into that? Obviously, PBPK model is not completely in silico. It is dependent on the in vitro, in vivo data. So that would be drug-dependent parameters such as protein binding, solubility, permeability properties, physiological parameters of the organ system such as blood flow, and anatomical parameters such as the volume of the tissue or organ that we are targeting. And ultimately, it combines those and studies the movement of the drug to generate this PK profile. PBPK models are widely used now in the regulatory applications as supportive information, and they're also instrumental in the early stages of product development, such as in candidate selection, be it a dosage form selection or even the molecule selection, because we can simulate the PK profile based on the molecular properties. They can help in clinical study design, dose selection, route extrapolation. How do we go from vaginal route to systemic or oral to vaginal? Any potential for drug-drug interactions can be studied, and most importantly, pharmacokinetics in special populations that may not be able to enroll in clinical studies, such as, for example, pregnant women or those with hepatic or renal problems. So we can study pharmacokinetics in all these populations in silico. So with that, I'll give it back to Lisa.
Sure. So we're going to share with you some of the work that we've been doing in the PBPK modeling area. Historically, I'm going to take you back to when we started some of this work. It was started under our FAME program. The FAME program was a U19 that was funded by NIAID. And in that program, the goal of that program was to develop vaginal films for HIV prevention. And within that respect, there were a range of products that were developed. They included small hydrophilic, hydrophobic molecules, as well as molecules that we knew were specific substrates for efflux transporters. So this provided a range of molecules that we could study. And the goal actually within this program, the FAME program, we were able to take molecules from the bench to the clinic. So in that case, if you think about it, we had beautiful sets of preclinical data, we had animal data, and we had clinical data. So we had those data sets that we needed to feed into the PBPK modeling. Next slide, please. So to this end, I'm in collaboration with Dr. Robert Bees at the University of Buffalo. Our team developed the first PBPK model for the female reproductive tract. And this model utilized the published data from the dapivirine vaginal ring that was available, and also our generated data from the dapivirine containing vaginally applied film that was developed within that FAME program that I just told you about. And that data was used to generate the clinical performance predictions and to simulate local and systemic drug exposure. And Siobhan, if you could click one more time for me. So there was a limitation of this very first model. So in this very first model, as you can see, the compartment that we focused on was the vaginal compartment. So we included the vaginal lumen, the epithelium, the stromal tissues, and the vaginal blood. But that's the specific compartment that was utilized in this initial PBPK model that was published as I show below. Now, in the figure in the upper right-hand corner, what we see is that figure demonstrates the accuracy of these predictions when compared against the clinical data. Now, this is the data for the dapivirine ring film. Now, as you can see, the black line is our predicted levels. The colored lines are the actual experimental data or the patient data from those studies. And you can see that the model had very good sensitivity with regard to its ability to predict and simulate. Now, Further, the figure in the bottom right actually shows predictions for the macaque model. So what we did was we actually modified the model to incorporate the key attributes from the pigtail macaque to perform predictions from the vaginal product performance in that model. So the anatomical parameters specific to the macaque were modified in that PBPK model. And so in that lower right-hand corner figure, what you're seeing is the data in the blue line is actually the predicted data. The dots are the experimental data. And once again, you can see that we had fairly good predictiveness with regard to what our experimental findings were. Next slide, please. Now, however, as we move forward and as we started to work with PBPK models for the female reproductive tract, we recognized that we had gaps in knowledge and gaps in data that we really needed to fill. The publicly available information that we could find with nice data sets were really not available to us. And so those were important to expand and reproduce and refine the model. We became very aware that surprisingly, there was a large lack of anatomical and physiological data for healthy non-pregnant women. For example, we knew blood flows in pregnant women, but we had none of that information for non-pregnant women. So we had to generate that initial data in ex vivo, in vitro, and clinical data. There also was a lack of information to describe the interplay between the drug product and the physiology of the female genital tract. So some of the things that Siobhan talked about become important here, like the release profiles, the mucoadhesion. And then finally, we had a lack of this in vivo data available in a single data set throughout the female reproductive tract to actually validate the model. So if you go to the next slide, Siobhan. So what we did was we started in our refinement of the model to expand these data sets and to fill these gaps that existed. So for example, some of this came into the preclinical space where we may have plasma protein binding available for some of the drugs that we're seeking to explore, but we need to understand what's the protein binding in the fluids, in the compartments, and the tissues in the compartments that we may be targeting. We also need to understand solubility in those compartments, as well as in our FAME program, we started to explore what are the relevant transporter metabolizing enzyme expression in the vagina and the cervix, but we had to expand that to the uterus, the fallopian tubes, and other compartments within the female reproductive tract, as well as understanding absorption and permeability rates in those other relevant tissues. We also recognized that we had to generate some clinical data to capture some of the anatomical and physiological parameters with respect to organ volume, blood flow measurements, and other aspects. Next slide, please. So this actually came through a series of clinical studies, which were the COSMIC studies. And those studies were led by Dr. Beatrice Chen and Catherine Chappell at the University of Pittsburgh Magee Women's Research Institute. So this is the first of the COSMIC studies that was conducted. In this COSMIC study, we actually collected vaginal fluid, pelvic ultrasounds, and biopsies of the vagina, cervix, and the endometrium. And from that vaginal fluid, the vaginal fluid was collected at screening. The women were actually given the choice that they could contribute whichever samples they chose, as many of the samples. We actually had almost 30 of the women that chose to give us all 4 pelvic ultrasound and all of the biopsy samples. So we had a really nice, robust set from this set of women. So from the vaginal fluid, we then had some of those physiological properties that we needed, the physicochemical properties. From the pelvic ultrasounds, we were able to get organ size and measurements, and also uterine artery blood flow measurements. And from the vaginal, cervical, and endometrial biopsies, we now had matched biopsies from a number of women. So we were able to really get important data relevant with respect to the enzymes and the transporter expression in female reproductive tract tissue. So next slide. So in summary, we really generated a significant amount of data regarding the female reproductive tract to develop and refine our initial PBPK model. So the model has now been expanded to include the oral compartment, the fallopian tubes, the uterus, as well as other compartments within the female reproductive tract. We've taken the data that we've gotten from ex vivo tissues, from in the clinic, with respect to organ dimensions and blood flow, and that's been able to expand and refine the capabilities of the model and the predictiveness. Separately, the work that we've been doing in vitro drug release with IUD systems in specific and other vaginal types of products has also been used to inform the model and parameters within the model. So this refined model now provides us a mechanism to enhance clinical study designs, to look at predictive levels of exposure in various areas within the female reproductive tract, which is important not only for targeting, but also from safety perspective. And also can be applied for development of next generation MPT products, and as well as to generate generic IUD product development has been used. So next slide, Sharon. So this has actually allowed us to create this work as a whole, a refined model. So we started off with our vaginal model through some funding from the FDA and the Gates Foundation. We've been able to expand into the entire female reproductive tract. And currently we're doing a pilot study, which is in the green box, that's funded by the Gates Foundation, where we're actually going in the clinic and collecting those fluids and tissues so that we can validate and refine the model and identify what is the predictiveness of the model. Can you click again, Siobhan? So simultaneously at the University of Dundee, they're doing similar data in animal models. So collectively, this data will really give us the data that we need in order to not only identify what the predictive nature of the model is, but also identify what are the key physical chemical parameters for drugs to get them to the target. So last slide. So just a couple key takeaways. We want to just reiterate that applying in vivo animal models for delivery performance evaluations is really critical to development of MPT products. These PBPK models we see having a very significant role, and also these collaborations with obstetrics and gynecologists and product developers really help us to make these tools so that we can advance these products. So just like to acknowledge all of the individuals that have been involved with the project, the people in our laboratory, Dr. Hillier, Dr. Chen in Chapel, and Carol Sprinkle from the clinics, Irvi and Corey doing some of the HIV work, Rob Bees and his team at the University of Buffalo, Dorothy Patton at the University of Washington, Pete Anderson, who's done a lot of the bioanalytical support for these projects, as well as the internet working with Population Council, Bridge Devlin, and Jeremy Nuttall for some of the dapivirine data that's been essential for this work to be completed. And also, of course, our funders. We really are lacking funding in the MPT arena, so we really greatly appreciate USAID, NIAID, NICHD, the Bill and Melinda Gates Foundation, and the FDA for their funding of this project. And we'll be happy to answer any questions.
Wow, thank you so much, Lisa and Siobhan. That was incredible. We want to thank you for sharing your time, your insights, the fascinating research, and I'm always impressed when you guys present with how much work you have done to really develop this. We're now going to move into our discussion portion of the webinar. Our hope is that this really sparks an engaging exchange. We encourage the audience to use this opportunity to share perspectives, ask questions, really learn more in depth from the experts that have presented today. We have a couple of questions already in our chat. Michael Santos, it looks like you were first. If you guys are able to share on video, that's great. If not, please just come off of mute. And Michael, I'll turn it over to you.
Okay. Thanks, Cheryl. Can people hear me?
Yes, thank you.
Oh yeah, well, I mean, thank you both for the presentation and IMPT for putting it on. Yeah, my question, in the empirical PK data you showed, I mean, I feel like, as is common for PK data, you just see a lot of inter-individual variability. And I apologize if I missed this, but I was wondering how well either of the modeling approaches you presented do in reproducing that observed variance, and then in particular, if maybe their ability to reproduce that lends any insight into the underlying physiological mechanisms that are responsible for the inter-individual variability.
Great, thanks for that question. I noticed that Rob's been chiming in a little bit on this. So Rob, do you want to jump in here and
Sure. So, I mean, that is an important concern. Capturing individual variability we are at least containing the observed variability with expected variability. We propagate this from all the in vitro experiments. We propagate this in terms of expected variance in the tissue sizes and permeation rates that were measured, for example, release rates. And so we do contain that. Now, in terms of estimating that variability more explicitly, one could refine the model with more of a simplified population, like a hierarchical nonlinear mixed effects kind of model. One of the challenges is having the repeated measures to do that. So there's sort of a trade-off there. One thing that one can do, and actually what our first paper with Lisa, I joined, so I was coming from a meeting with the provost, so
yeah.
I was late. So there was a reference to the ReproSim model. So the model through sort of the uterine space is currently under review, and as soon as it's accepted, we're going to post that to GitHub, and then you can use that on your own to explore these types of questions, mainly for simulation. But you can adjust the variability, you can adjust population types, you can adjust the presence or absence of different transport type of processes, and that's implemented in something called MRG, something called MRG Solve. It runs in R and C++. We have a second paper that has the graphical user interface, where you can actually we have sliders that you can actually explore very specific parameters and variabilities in a much more user-friendly way. And that will allow you to at least evaluate where though the variability may be arising. For all these models, we've done both local and global sensitivity analyses, and they will be part, of course, of the main publication. But what we see is that depending on the route of administration and the biophase of interest, the processes that are likely to contribute to observed variability are different. So you can get a sense of what you might need to optimize, or what you might need to know really, really well in order to have some confidence in those predictions. I do hope that addresses the question, or if I missed it, please, I'm happy to receive clarifying comments to refine the answer.
Yeah, thanks. That's very helpful. I guess the kind of second phase of my question is, whether, if through the modeling you can get some insight into the mechanisms, which, as you say, I mean, makes sense. They the mechanisms vary depending on route of administration, other kinds of factors. But is there anything that can be learned that can then feed back into product development? I mean, I was noting, if I remember the kind of sequence that you showed when you showed, your 3 different attempts in the product development, you were really trying to boost the exposures, not because you're trying to boost the exposures of the most exposed individuals, but presumably because you're trying to boost the exposures of the least exposed individuals, but recognizing that there's just this really wide range. So presumably if something could tighten that range, then you could also get away with less drug, for example,
Right.
in the product, with various advantages for cost and safety and things. So yeah, I guess the second part of it was, well, if you can't understand something about the mechanism, are there any levers in product development that you can use to kind of offset the variability?
Yeah, so just to going back to the example I showed, so that was the non-compartmental analysis, and it's really valuable because it's very quick. You get some insights into how we can change things on the formulation side to that can impact those very simple parameters such as Cmax, AUCs, or THAVs, right? However, the limitation there is that you need to have really less variable data to even develop a model, something like that. So in order to be confident about that Cmax or THAV, your data itself should be less variable. However, like I think as Rob mentioned, for PBPK, we have that. I think we have more opportunities to sort of, non-scientifically speaking, tweaking some of these parameters and seeing the impact on the pharmacokinetic profile, and we could choose the best one that makes sense. For example, your release might be optimal, but your permeability into the tissue might not be. And what could we do from a formulation standpoint to change that? Now, we have not explored that in our work, but we could theoretically incorporate some permeation enhancers. So I think with PBPK model, there is more opportunities to learn about the mechanisms. Empirical models, not so much.
Great, thank you.
Thank you. What an incredible conversation. Purna Shah, I see you have some questions that you and Robert are discussing. Would you like to come off mute and use this opportunity to address them?
Yeah,
Wonderful.
great presentation. It was very interesting. Thanks. Rob already answered half of my question. The reason behind I asked that question is because when we talk about female reproductive system, and especially vagina, where we don't know the volume of cervicovaginal mucus, and it varies from woman to woman. So when we are looking at, for example, dosing of those drugs, which we don't expect them to cross or permeate into the bloodstream. So during that time when we are working on the dosing strategies of how much we should add, because at that time the drug will also, for example, when you're talking about contraceptive, the drug will also be diluted in the presence of semen. So when we have to decide the dosing strategies, how well the model systems work and how more we should estimate our loading. For example, let's say the films or the rings or different type of system.
You're absolutely right. These are all factors that will contribute and factors that we need to explore further. And Rob and I have had long discussions about how to incorporate each of these into the model. Rob, I'll let you add to that.
Sure. No, I was saying basically the same thing. So you can vary those both volumes, and you can vary the characteristics in terms of access to tissue or what have you, and get it, and turnover rates and that sort of thing. The issue is getting parameters that are actually a true physiological reflection of what you're observing. So the measurement. So one of the critical things is that we can do this in the model, but then Lisa and Siobhan are able to actually do the experiments and make the direct measurements that have informed it. So the model continues to evolve to allow this. Currently, we have a relative, the model's relatively static in terms of fluid volumes and characteristics. So with one caveat, the models can be completely customized. You can grab the ordinary differential equations, we have the scaffolding that you can make those changes. But what we have for example, in our graphical user interface is somewhat more limited, just because there are so many features that that you could vary. But we could, that again can be customized. This will all be open source eventually. And you can explore it, but one of the critical thing is you can do whatever mathematical simulation you want to do, especially if it's something new, but there's no substitute for experimental confirmation for refining the model, increasing your confidence in this. And so you end up with sort of certain spaces of operating conditions, right? Characteristics where you have confidence. So if you fall within this particular space, you say, okay, well, I think the model will give us predictions that we can have a little more confidence than a place where we haven't done those types of measurements, but it maybe gives you a place to start and helps you guide
Right.
what that first experiment may be. Oh, Lisa, oh, sorry, go ahead, please.
No, yeah, that makes a lot of sense. That it helps to get us started.
Wonderful. We're incredibly grateful, Robert Buys, to have you on board, another expert in the leading field. I don't see any more questions in the chat before I take some of my questions. Robert, I'd love to open up the floor if there's anything that you would like to expand on in this field, or directly tying it to how these models can then be tied to the MPT field and the complexities.
So we've actually had some experience with modeling MPTs, mainly the data coming from I guess what Lisa, correct me if I'm wrong, but I think it's mainly from the matrix experience, and now we're moving that direction with some of the Gates work, I believe.
And Rob,
sorry,
Siobhan also presented, you may not have been here, some of the latch data.
Oh, yes, of course, yes,
yeah.
the EFTA levonorgestrel.
So you've you've been working with all of those programs.
Yes, and we've we when we were evolving the models, for example, we generally there was some not surprising, but there were definitely refinements that had to be made in terms of being able to capture the connection between release profiles and uptake and plasma concentrations, local concentrations. In particular, we have quite a highly refined model, for example, for levonorgestrel release in the uterine space and uptake and those concentrations. They were consistent when we moved into the vaginal space, for example, with latch. There were some additional refinements that we had to undertake. Whereas the model translated almost perfectly for EFTA and for dapivirine. But I think so what that means is that now we have those sets of characteristics, so that for an MPT with molecules with those characteristics or similar characteristics, we have some reasonable confidence in the prediction. So you can take the in vitro release rates, for example, that are measured in Siobhan and Lisa's lab, and incorporate them and get a reasonable prediction of both local and systemic concentrations. I don't know, Lisa, and also Cheryl, if this is addressing really the core of the question that you raised, and these are living models. Eventually they'll be on GitHub, so you can grab them and refine them and use them, and they'll be tracked. So hopefully, folks can they contribute to sort of the whole ecosystem of knowledge here. So.
Yeah, thank you so much.
Rob, that's perfect, because we were talking about challenges specifically from the MPT, and you really honed in on some of the challenges that we faced with each of these different projects.
Wonderful. Are there any other questions? If not, I'm going to ask the hard magical question about, I understand that these models are now given to FDA or regulatory approval bodies as supplemental information. Do you see that expanding, and how do you think that they may look upon this as more data comes? Do you think it will be more acceptable moving forward versus animal models, or animal models always going to continue to be something that needs to be presented alongside of this?
Well, I know, at least from our collaboration with FDA and ongoing conversations in a not as a submitting party, but as a collaborating party, there's a great deal of interest in leveraging these types of models to answer questions, especially for long-acting products in this space, and whether or not these approaches could be used to reduce numbers of experiments or facilitate the efficiency of a development path. But I can't comment exactly on how the FDA would view, so even if
Of course.
view these things. But there certainly was a great deal of interest, and the FDA regularly engages in submissions that have a great deal of model informed drug development that reduce numbers of experiments. I think the space for the regional administration there's less experience on the part of the regulators there, and I think the original stimulus or impetus was for the broad agency application funding that we received was how can these approaches be leveraged when you have these 5-7 year products where other interests in terms of entering that market are very, very difficult to it's very difficult to get there. Can we use these approaches so that you don't have to run an experiment for that length of time? I don't know, Lisa, please, if I'm or Siobhan.
No, I agree, Rob. They're of significant interest as we've seen with the funding mechanisms that have come from FDA, as well as collaborations that have been generated. And as we move into this realm of where FDA wants to pull back on some of the animal data, especially when we think about monoclonal antibodies and other things, these data will become more and more important. And Siobhan actually has some direct experience with, how this model's facilitated applications to move forward. Siobhan, if you want to talk a little bit about that.
Yeah, I think on the experimental modeling side, I think Cheryl, as you mentioned, in the near future, at least, I think animal models are going to stay. I think they are the mainstay for both PK as well as safety, right? But I want to go back a little bit from on the MPT space. Today we discussed vaginal MPTs and specifically HIV prevention and contraception, but there are MPTs that are purely STI based and by other routes of administration, oral, for instance. I think the acceptance of PBPK modeling approaches from some of these other routes is I think there's more evidence of it from the regulatory side. FDA, as well as I think there are some guidances from EMA, WHO has also put out a few things out there. So I do think that when we are, as a low-hanging fruit for MPT, people working in this field, I think there is applicability for PBPK models when you're talking about more established routes of administration. For something like this, I think it can be a really great supportive data at this point, but I think we are moving into that direction, and hopefully, I know Rob doesn't like the word validate, but when the model is fully out there, maybe there is more acceptance from the regulatory bodies as well.
Oh, thank you so much. This is an incredible amount of work. We're excited to have all three of you. We couldn't get three better experts on this field to join us. So thank you. Claudia, I'm going to hand it back to you really quick to wrap us up.
Thanks, Cheryl. I just want to take a moment to extend our sincere gratitude to Drs. Lisa and Siobhan for such an insightful and informative presentation. We truly appreciate the time and expertise you shared with us today. A special thank you to NICHD, whose generous support makes all of this possible. Before you jump off this call, we just want you to know that we'd love to stay connected. So go over to www.impt.org, sign up for our listserv, and you'll be the first to know about upcoming events. For example, our next Let's Talk MPTs webinar, which will take place on September 4th, same time, 8 a.m. Pacific time, and we'll be focusing on the dual prevention pill with speakers from AVAC, Population Council, and the IMPT. Now I'm done. Thank you again for joining us, and we wish you a wonderful day or evening wherever you are. Goodbye.
Thank you. Thank you for the opportunity.
Yes, thank you very much for the
Thank you, everyone.